Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So, in this particular lecture, I will be explaining you important regulatory steps in glycolysis. Let's look into the details. So, there are three irreversible steps in glycolysis, and these three irreversible steps are the ones that are regulated in glycolysis. And those steps are hexokinase glucokinase step, phosphofructokinase 1 reaction, and pyruvate kinase reaction. The main regulatory enzyme out of these three main regulatory enzyme of glycolysis is phosphofructokinase 1, which is simply abbreviated as PFK1. In fact, PFK1 is referred as master controller of glycolysis. Let's look into the details. Regulation of glycolysis at hexokinase and glucokinase step. So as you all know, hexokinase, it is going to convert glucose into glucose 6-phosphate, very first step in glycolysis, which is going on in most of the peripheral tissues using hexokinase as the enzyme, except in the beta cells of pancreas and in the liver, where glucokinase is the enzyme for the first step in the glycolysis in these two tissues. Now, hexokinase, it is feedback inhibited by its own end product, that is glucose 6-phosphate. So, whenever glucose 6-phosphate is accumulating, it will have a feedback negative effect on hexokinase, thereby slowing down that reaction. Now, glucokinase do not have similar feedback effect by glucose 6-phosphate because glucokinase is present in the liver, and liver has to consume excess glucose that is there in the blood and allow that glucose to run into glycolysis, and thereby the glycolytic intermediates has to go into different metabolic fate, different metabolic pathways so that glucose can be used. So that's why glucokinase, which is there in the liver and also there in the beta cells of pancreas, do not undergo negative feedback inhibition by end product glucose 6-phosphate. However, glucokinase is induced by insulin, but that kind of induction you don't see for hexokinase, because whenever the person is in well-fed condition for a long period of time, when the blood glucose levels are building up, at that time, so the glucose is going to release insulin from the beta cells of pancreas, and that insulin while binding to the hepatocyte membrane, so it is going to induce expression of glucokinase gene, thereby making more and more glucokinase enzyme available, so that glucose that is there in the blood, it is taken out of the blood and used for metabolic reactions, thereby it is controlling the blood glucose level. That's why whenever person is maintaining himself or herself in well-fed condition for a long period of time, whenever glucose is building up in the blood, so insulin will make sure to induce glucokinase gene in the liver, and thereby expressing the gene for glucokinase and increasing the synthesis of glucokinase there. Now let's move on to see a most important regulatory step in glycolysis, which is done by phosphofructokinase number 1, which is PFK1. Now the PFK1 is the main regulatory enzyme for glycolysis. So it has negative modulators and it has a positive modulators. Now the negative modulator for PFK1 is ATP. Whenever there is a rise in ATP levels in the cytoplasm, it simply indicates there is excess energy in the system, and that's why ATP acts as a negative modulator on PFK1, thereby it is going to decrease the reaction. So thereby glycolysis need not go on at a higher level. Whereas AMP, whenever there is accumulation of AMP in the tissues, so AMP acts as a positive modulator on PFK1, because presence of AMP indicates lack of energy in the system. That means glycolysis has to go on at a higher level, and that's why AMP acts as a positive modulator on PFK1. Especially in the skeletal muscle, you can observe this. Whenever person is doing exercise, AMP level increases, and that AMP is going to keep glycolysis at higher level by keeping PFK1 active. Citrate. Citrate acts as a negative modulator on PFK1 because presence of citrate in the cytoplasm simply indicates high energy status of the body, and that's why citrate will have a negative effect on glycolysis. And all these molecules that I have explained right just now, ATP, AMP and citrate, all these molecules, they will have their positive and negative effect on PFK1 accordingly in other peripheral cells except liver. So, presence of ATP, AMP and citrate do not have any effect in the liver PFK1. PFK1 present in the hepatocyte do not have much effect from ATP, AMP and citrate. So, it is only for other tissues. What is the molecule that regulate PFK1 activity in the liver? It is fructose 2,6-bisphosphate levels. So, the concentration of fructose 2,6-bisphosphate in the liver is the main controller of PFK1 activity. So, let's see which molecule or which enzyme is or what are the hormones that regulate fructose 2,6-bisphosphate levels in the liver. Now, the fructose 2,6-bisphosphate levels that control PFK1 activity, it depends on the cellular metabolic status. Whenever person is there in fasting condition, whenever there is a low blood glucose level, low blood sugar level, as it is shown here in the figure. So, there will be in response to low blood sugar, there will be increase in glucagon released from alpha cells of pancreas. And what this glucagon does? Glucagon will bind to glucagon receptors present over the hepatocytes, and glucagon receptor activating adenylate cyclase, cyclic AMP increases, and that will increase protein kinase A enzyme. And this protein kinase A enzyme is going to phosphorylate an enzyme called PFK2. And this PFK2 is a bifunctional enzyme. It has got two enzymatic subunits. One is PFK2, that is kinase subunit. Other is fructose bisphosphatase 2, that is the phosphatase subunit. Just to simply put it as the two subunits in PFK2 that is shown here is kinase subunit and phosphatase subunit. So, PFK2 enzyme, it becomes inactive whenever it is phosphorylated, because glucagon, by activating protein kinase A, it is going to phosphorylate PFK2, meaning it will add phosphate over the surface of PFK2. At that time, the kinase subunit of PFK2 becomes inactive and the phosphatase subunit becomes active. And what is the role of PFK2 phosphatase role? So, the PFK2 phosphatase subunit, what it does, it is going to take fructose 2,6-bisphosphate, which is there in the cytoplasm, back into fructose 6-phosphate molecule. So, fructose 2,6-bisphosphate will go back into fructose 6-phosphate. That means the concentration of fructose 2,6-bisphosphate decreases in the cytoplasm, and whenever there is a decrease in the concentration of fructose 2,6-bisphosphate, its positive effect on PFK1 is not there. That means it is going to decrease glycolysis, and the antagonistic pathway to glycolysis is gluconeogenesis. So, gluconeogenesis becomes more active whenever glycolysis is decreased, and that's what glucagon does. So, glucagon under fasting condition, in response to low blood glucose level, by increasing protein kinase A activity, it is going to keep PFK2 phosphatase subunit active, thereby fructose 2,6-bisphosphate is taken back into fructose 6-phosphate, and that means glycolysis decreases because its positive effect of fructose 2,6-bisphosphate is not there, and the gluconeogenesis increases there, because the fructose 2,6-bisphosphate concentration also has an effect on gluconeogenesis. Whenever fructose 2,6-bisphosphate concentration is high, as I told you before, PFK1 activity is increasing and thereby glycolysis increases. At the same time, gluconeogenesis, which is an antagonistic pathway, it will decrease. Now, what glucagon does? Glucagon, by keeping protein kinase A active, thereby increasing the activity of PFK2 phosphatase, it is going to take fructose 2,6-bisphosphate back into fructose 6-phosphate, thereby decreasing the concentration of fructose 2,6-bisphosphate, thereby decreasing glycolysis and increasing gluconeogenesis to maintain the blood glucose level. Now, let's see the opposite effect. Whenever person goes into fed condition where blood glucose level rises, a high blood sugar. So the high blood sugar releases insulin from beta cells of pancreas, and what this insulin does, insulin is going to activate protein phosphatase number 1. And this protein phosphatase number 1, as the name says, it is going to remove phosphate from PFK2 surface. That means once the phosphate is taken out of the PFK2 surface, so PFK2 kinase become active, PFK2 phosphatase become inactive. So when the PFK2 kinase is active, what it does? It is going to convert fructose 6-phosphate into fructose 2,6-bisphosphate. That means fructose 2,6-bisphosphate concentration rising, and that fructose 2,6-bisphosphate is going to keep PFK1 active. At the same time, it is going to keep a gluconeogenic enzyme, that is fructose and 6-bisphosphatase, inactive. So because of this, so what insulin do? By keeping PFK2 kinase active, it is going to increase fructose 2,6-bisphosphate concentration, which is a positive modulator on PFK1, thereby increasing the glycolysis. And also fructose 2,6-bisphosphate is a negative modulator on gluconeogenesis and decreasing the gluconeogenesis. And that makes sense. Why? Because when the person is in fed condition, glycolysis has to go on at higher rate, gluconeogenesis has to go down, and that will be maintained by maintaining higher concentration of fructose 2,6-bisphosphate, and that is done via keeping PFK2 kinase active. So what happens under fasting condition? Under fasting condition, low blood glucose, glucagon increased, protein kinase activated, and that protein kinase is going to phosphorylate PFK2, and phosphorylated form of PFK2, it's a phosphatase component is activated, and it's going to decrease fructose 2,6-bisphosphate bisphosphate levels in the cytoplasm, and decreased levels of this molecule will keep glycolysis low, gluconeogenesis high. That means blood glucose level is maintained under fasting condition mediated by glucagon. And this is how glycolysis and gluconeogenesis, they are regulated simultaneously by a single molecule, and that is fructose 2,6-bisphosphate concentration. Now, other than liver, muscles and cardiac and other tissues, they got an isoenzymes of PFK2 and pyruvate kinase are refractory to inhibition by phosphorylation mediated by protein kinase A molecule. Thus, muscles and other tissues, glycolysis is not altered in the presence of glucagon and epinephrine. So here, allosteric modulators of phosphofructokinase 1 is given. On the left side, you see the activators of PFK1. On the right side, you see the negative allosteric modulators, which is referred here as inhibitors of PFK1. So AMP, fructose 2,6-bisphosphate, ADP, potassium, phosphate, all these molecules act as a positive allosteric modulators, while ATP, citrate, calcium, magnesium and low pH, they all act as a negative modulators on PFK1. Now let's look at regulation over pyruvate kinase enzyme. Now the pyruvate kinase enzyme, it is one of the regulatory enzyme in the glycolysis, which is running the last step in glycolysis, conversion of phosphoenol pyruvate into pyruvate. So the fructose 1,6-bisphosphate, which is the intermediate of glycolysis, way ahead of where pyruvate kinase is working in the glycolysis. So fructose 1,6-bisphosphate, it has got a feed forward activation on pyruvate kinase. Now the higher energy status, that is ATP, acetyl-CoA, presence of acetyl-CoA in the cytoplasm indicates higher energy status. Presence of long chain fatty acids in the cytoplasm indicate that gluconeogenesis has to go on. And all these things will have a negative effect on pyruvate kinase enzyme. And also note that pyruvate kinase enzyme is active whenever it is in dephosphorylated condition. So insulin, when person is in well-fed condition, what insulin does? Insulin is going to keep this pyruvate kinase in a dephosphorylated state by removing phosphate, because insulin is going to activate protein phosphatase enzyme, and that is written as PP here. And this protein phosphatase, it is going to remove phosphate from the surface of pyruvate kinase and keep the enzyme active, thereby glycolysis increases. And whenever person goes into fasting condition, so glucagon is going to activate protein kinase A enzyme, and that protein kinase A enzyme is going to add phosphate to the pyruvate kinase and keep the enzyme inactive, thereby glycolysis is kind of inhibited. So, insulin under well-fed condition, it is going to keep glycolysis active by keeping pyruvate kinase in dephosphorylated state, whereas glucagon is going to keep glycolysis low by phosphorylating pyruvate kinase. This is how insulin and glucagon act on glycolysis apart from their action on phosphofructokinase 1 via phosphofructokinase 2 by maintaining the concentration of fructose to 6-bisphosphate levels. So that's about regulation of glycolysis on 3 important reactions, and that is hexokinase glucokinase reaction, PFK1 reaction and pyruvate kinase reaction. Thank you.